Hey science fans, welcome along. So today we're going to look at enzyme action and the required practical for AQA biology triple trilogy. Everyone needs to do this one. So this is testing how different conditions affect the rate of enzyme action. Now, enzymes are biological catalysts found within your body. They are made of protein, which are made of amino acids, and they enable us to have biological reactions and speed them up. And we're going to look at the different range of pHs today that can affect the rate of their action. Remember, rate is speed, how much they produce their product divided by time. Let's do it. I'm doing the practical where I'm looking at how different pHs affect enzyme action, and what I'm looking for is the small window, the optimum pH for my enzyme action. If it's either one of those, then we'll get denaturing. Either side of that optimum will get denaturing of the enzyme, which means that the active site will become deformed and no substrate can fit into the active site, which means it will no longer work. I'm going to be using amylase. Amylase is the same enzyme that you produce in your salivary glands. Okay, so this is our investigation. So we are investigating the effective pH on the rate of reaction of amylase enzymes. So the effective pH, so we're going to be changing this. This is my independent variable. The rate of reaction is what I'm going to be measuring, so this is going to be my dependent variable. Now, I'm going to use amylase to break down starch. So just so we remember, starch gets broken down into glucose. Starch is insoluble, so it means we can't dissolve that into our bloodstream. Glucose is soluble, so it means that we can dissolve that into our bloodstream, take it to every single cell and use it for respiration. Amylase is the enzyme and starch is the substrate. So together what I'm expecting is the amylase enzyme is going to take the molecule of starch and then it's going to form an enzyme substrate complex, and then it's going to break it into smaller glucose molecules that can then dissolve into your blood and we can use. We're going to use different pH solutions. These are called buffer solutions. We're going to use a water bath so that they can be kept at a constant temperature. Now, we can do another reaction where we look at how temperature affects pH, and there is an optimum range or an optimum temperature, and either side we get denaturing or a slow rate of reaction, but that's a different one altogether. We're going to use continuous sampling, so I'm constantly going to be checking the enzyme action over time. And we're going to be using iodine as our indicator, because we know that iodine from our food test goes black in the presence of starch, but it doesn't in the presence of glucose. It just sort of stays that kind of browny colour. And I'm going to use that. So I know that my starch has been broken down into glucose when my iodine no longer goes black. So let's have a look at the equipment. This is a useful piece of equipment. This is called a water bath, and it's going to maintain a constant temperature of my reactants. I'm going to set it for 35 degrees because I know that amylase works really well at body temperature, and it's going to be working at a pretty good rate of reaction. If it was too low, my enzyme would be working too slowly. So it's a good kind of link to chemistry there. We'd have a slower rate of reaction because the collisions would be lower. And then if it was too high, we would be deforming the active site, and so my enzymes
popping them into 4 different pH solutions. These are called my buffer solutions, so they maintain that pH throughout.
What I'm going to do now is put 4 test tubes in with 4 centimetres of starch and 4 test tubes in with 2 centimetres of buffer solution. Going to label them all up, obviously. Let's go. You'll notice I didn't use a measuring cylinder for that. I used a pipette, pasta pipette, and that pipette measures 2 centimetres every time you do a squeeze. So it's not as accurate as using a measuring cylinder or a balloon pipette, but it measures the same proportion. It can only measure 2 centimetres each time. So 2 of the starch, 1 of each of the buffer solutions. So now that's just getting up temperature. I'm just going to measure the amylase, and that is going to be 10 centimetres in a tube in there as well, separate. Just going to wait for them to get up to 35 degrees and then we're going to crack on. Beauty about a water bath is everything will maintain that temperature And while I'm waiting for it to reach that temperature, I'm going to use this dimple tray and I'm going to put 1 drop of iodine solution in each dimple. Now the iodine is going to be my indicator as to whether the reaction is complete, whether the enzyme has broken down the substrate into glucose, because if it does, it won't change colour. So I'm just going to do that while I wait. Now comes the bit where I've got to work quite fast. So I'm going to put 2 centimetres of my buffer solution in with my starch, and then I am going to put in 2 centimetres of my amylase. As soon as I do that, I've got to stir, 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 start my stop clock, and every 10 seconds I am going to put a drop of my amylase solution, or my amylase and substrate solution, into the dimple tray. And when it stops producing that blue-black colour, I know the reaction is complete. Let's go. That one worked in 1 minute and 48 seconds. That's really good. Worth noting, you might need 2 dimple trays for the different varying pHs already and set up, ready to go. Mine fortunately worked with 3 spaces to go, so that's really cool. Let's have a look at the rest of the results. Very rough table here. So I've got the pH of solution, the time taken for amylase to completely break down starch at 35 degrees C, and that's measured in seconds. Here's an example of the enzyme action. So I started here, and then I went up in rows, and you can see these two, no change. So therefore all of the glucose has been, all of the starch has been digested. Boom. Remembering our practical is to do with the rate of enzyme activity. So I actually need to convert this time into a rate. So the amount of enzyme or product used or product produced divided by time. So I'm going to do that to work out, it's basically going to be moles per minute, moles per second, sorry. So what I'm going to do is I'm going to do 1 divided by the time, and that is going to give me a rate that I can actually plot on a graph. Let's go. So I've got all my rates of reactions, and all I've done is done 1 divided by the time taken, because we use the same volume, so we're going to say it's 1 mole of enzyme product, 1 mole of enzyme reactant used, or 1 mole of product created divided by time, because we use the same volume on each. So I did 1 divided by this, and it's given me these to create a nice set of results. Now obviously the faster it goes, the greater the rate of reaction. That means that we will get a graph that looks like this in the right place. So we can say, yep, that's the optimum because the rate is higher. If we just plotted the times, it would still look like this, but it would look like the one that was working quickest was actually the poorest in terms of its rate. So that's why we convert it into rate reaction. So I just need to plot a good graph to enable us to use to show how the rate of enzyme activity is affected by the pH. Independent variable goes on the x-axis, the rate of reaction goes on the y-axis, so I'm just going to do that now.
Right, so here's my graph, which doesn't look complete, which of course it's not at this point. So I put the rate of reaction there in U moles per second, and I put the pH along the bottom. Now what this tells me is, of my results, pH 5 was the optimum. So I would have to, if I was to extend this, I should be working on looking at pH 3 and 4, because we can see there's a clear decline there. What I would expect is to see something that looks like this. I'm going to draw this on with a pen. I would expect to see something that looks like this. Okay? I haven't seen that, that's not what I've got, so I'm only going to plot what I've got, but we know that the optimum is going to be around here somewhere. So I would, if I was to extend mine, I would look at doing pH 2, 3 and 4. I would also look at doing something that was about pH 4.5 and 5.5 to see if either of those were more optimum. There's a very narrow range at which enzymes work at in their optimum, because otherwise the active site just denatures. It's the same for temperature, very narrow range. It's around 37, 40-ish, and again we would do them closer. So if you were doing this for temperature, you do exactly the same practical, except you would have different temperatures along the bottom, and you would need to extend or to make your results more accurate, do more data around those points, do closer intervals. Okay, I haven't given this a title, but this is a graph to show the rate of reaction of the enzyme amylase at different pHs. That's it. Boom. you need to be able to give the method, the different variables. You need to be able to apply it to different enzymes. So they might give you something like pepsin or renin. They could give you anything, but they will give you enough information so that you can sort of have a look at writing a method that is similar to this. So remember, we always use measuring cylinders to measure volumes, pipettes for smaller volumes, and they will normally give you what the indicator is, but just work off the back of this one. They could ask you about temperatures, and again, just work off the back of this one. They always ask about water baths, because it maintains this constant temperature. You can make a water bath using a Bunsen burner and just a beaker of water. Not nearly as accurate though. You need to think about, are your results reproducible? Could someone else do this practical and get valid results? Is it repeatable? Could you do it again and get valid results? Have you taken precise measurements? Is there a way of taking more precise measurements? That kind of thing. So lots of discussive points, but make sure you learn this method so that you can just reel it out and all the different variables and how you control those variables. Done.